Dear moderator, dear colleagues, dear students and guests of our university, there is my big pleasure to present the results of my research groups titled Adverse Effect of Ambient Particular Air Pollution on Gut Microbiome, Translational Research. Okay. Doesn't work. Okay. Okay. It's a little bit provocative question. Why is microbiota and how it's connected to ambient pollution, and why we pick up this line of research. I would like to point it out that my background is not microbiologist. I'm the physiologist. I'm graduated from the Department of Physiology, Human and Animals. Unfortunately, this department doesn't exist anymore in the Institute of Biology and Medicine, but I'm graduated from this department, and my background in physiology allowed me to see the broad context in any problem and understand that all research in microbiology, in molecular biology, in biochemistry connected overall to their health of their human. And as a confirmation of this, and taking into account that my presentation is the first one in during the plenary session of this conference, I would like to give the short history overview. And on this slide, you can see the slide from the review article offered by Berg, Rybakova, and Fisher, published in Microbiome Journal in 2020. And it's really a very good article which described the history of microbiome research from 17th century until our days. And you can see how the new development of new techniques and equipment can boost the microbiological research. And we moved from 17th century up to 21st century from understanding on the paradigm of microbes as an unsocial organism causing disease, like infection disease, and mostly in early days, the researchers, of course, study the role of microorganism as a cause of disease, to the holistic view of microorganism being the center of one health concept, positively interconnecting all areas of our life. And on this slide, you can see the overview, and for us, it's like real everyday practice. We don't think it doesn't surprise us anymore that human is a supraorganism composed of mixtures of host and microbial cells, genes, and metabolic attributes. And we rely on our microbiota to digest, to produce vitamins, to shape our immune system. And we can see that microbiota distributed on the skin, respiratory tract, in oral cavity, and of course in gut. And here you can see the main predominant bacteria genera in these habitats in our organism. Of course, we live in the real environment, and we eat every day. We take medications, and what we eat, what is our lifestyle, all of these significantly affect our microbiota. And on this slide, you can see the picture from the Frontiers System Biology review in 2022, how modernization affected the composition of our microbiota. And of course, this urban lifestyle was affected our microbiota, and as a consequences of this, of our everyday routine, we can see the development of bacteria community composition imbalance implicated in the pathogenesis of several intraintestinal, such as inflammatory bowel disease, e.g. Crohn's disease and ulcerative colitis, as well as extra-intestinal disorders, such as diabetes, cognitive deficiencies, and hypertension. Since traditional factors such as diet, medication, socioeconomic status, and gender only explain 16% of inter-individual variation in the intestinal bacteria. Intestinal bacteria composition, the question arises whether environmental factors such as air pollution, exposure might play a role. And this is their quick answer why my talk focused on the connection between their ambient air pollution and the microbiota. And also talking about the trends in microbiology and using the artificial intelligence also in research of microbiology, especially their implication of machine learning-based models for the prediction of disease, we see the significance of our understanding and precise understanding of the microbiome and changes in the composition of microbe for the prediction of disease. In this articles, which is published in Trends in Microbiology in July 2024, we can see that the human microbiome has been increasingly recognized as having potential use for disease prediction. And author said that the most promising improvement in microbiome augmented disease prediction are seen for liver disease, respiratory disease, diabetes mellitus, inflammatory bowel disease, in neuropsychiatric conditions. As improved, the prediction relies on accurate microbiome definition, detection, and diversity metrics. Important considerations during bioinformatics and experimental design must be taken into consideration to limit the effect of unwanted technical bias. And I would say, taking into account that we have a lot of young scientists and students in this auditorium, that machine learning, this is implementation of machine learning in the research is the future, and you have to take this into consideration and apply this knowledge to this field as well. And from this study, we understand that the significance of and necessities of the research in the field of the microbiome, and especially how the more precise understanding of the role of different kinds of bacteria in the etiology of different diseases, as well as a search for new approaches for microbiota correction is the current topic for research, not only for the researchers in the, not only for the microbiologists, but these tools can be used for all disciplines to their medical care. And moving forward, I would like to connect the significance of microbiota for the human being as a supra-organism and the health of how the organism with the ambient particle pollution. And here on this slide, you can see the map. And on this map, you can see by the more intensive color, the number of deaths in different parts of the world because of the air pollution. And according to the world data from World Health Organization, 9 out of 10 people breathe air that does not meet World Health Organization pollution limits. And air pollutants include gases and particulate matter, and collectively are responsible for 8 million annual deaths. World Health Organization working on the estimation on the allowance of the pollutants in the air. And on this table, I hope you can see it clearly. You can see the list of the 6 so-called common air pollutants, such as particle matter, PM, ozone, nitrogen dioxide, sulfur dioxide, and carbon monoxide. Here you can see the table and difference, the global air quality guidelines from 2005 and 2021. And if you compare the allowance from World Health Organization between these 2 years, 2005 and 2021, you can see that updated values provide clear evidence that poor air quality damages human health at even lower concentration than they are assumed previously. You can see the data for the PM 2.5, particle matter PM 2.5. Air pollution is one of the most prevalent airborne factors affecting global public health. And you can see here then, for example, in 2005, according the data of World Health Organization, the threshold was 25 grams per meter. But in 2021, you can see that the significant decrease, 15 microgram per cubic meter. So it means that we are more prone to the effect of pollutant in the air, and this connected to the development of different type of disease. Moreover, the guidelines also highlight the importance of different particulate matter types, for example, black carbon, elemental carbon,